Good day to everyone. Welcome to another program of Coffee Break. And today we are going to discuss about the COVID-19 variants over the globe. There are so many variants prevailing all over the world at the moment, and the COVID-19 variants cause more infectivity and more transmissibility, and it spreads faster and cause more damages all over the globe. So it's worth discussing about the variants and how it happens and what are the consequences of the variants of a virus. It is calculated and mentioned that there could be a generation of a variant, two new variants each month. But at the moment, there are three variants which are raising concern all over the world. Let's see what they are and how do they act. Now I'm going to start from the beginning and explain you how these viruses act on the host cells and how do they get mutated. To understand this properly, we have to know the basic molecular biology sciences. In a very brief explanation, I will start from DNA and RNA and explain and revise your knowledge about the molecular biology regarding the structure of them. This is a gene, and this contains DNA. If we enlarge one of these parts properly in this gene, there are double helicals, as the students, scientists and doctors know, double helicals of nucleic acid formation, and these double helicals are consistent with sugar phosphate backbones and various bases connected with each base with hydrogen bonds and make this double helical. Usually in a DNA, adenine, thymine, guanine and cytosine are the bases, so they join these places, and usually adenine with thymine, guanine with cytosine. So this is the DNA helical, and in an RNA helical you have the sugar phosphate backbone, and you have only one strand. This is called single-stranded RNA. In this, you have the sugar bases. This is the sugar phosphate backbone, and these bases are a little bit different from DNA. Adenine, uracil, guanine and cytosine. So these bases are in various patterns throughout this genome, and that sequence make the particular genome. For an example, you have 20 essential amino acids in our human animal genomes, and when these bases connect together, they form one of these amino acids. If this base pattern changes from one generation to the other generation, then a mutation arises. For an example, if you have adenine, uracil, guanine, cytosine here in this generation, and when it goes to the other generation, instead of this adenine, if cytosine comes to this place and adenine goes to this place and cytosine goes to this place by the constant battle of the virus replication with the host cell, this becomes a variant and it causes a new mutation. So this is the basis of mutations in a virus. A virus is not going to be mutated while it is not infected the host cells. The mutation happens with the constant battle in the mutation, in the replication of the viral inside the host cells. Especially if this base pattern changes, that change makes completely different essential amino acid in that place. So it is a different variant of that species. Now we got an idea about the difference between the DNA helical and the RNA single strand. So our COVID-19 virus is a single-stranded RNA virus, and let's see how does this genome behave. This single-stranded RNA, if we take it like that, it contains 30,000 units bases throughout this genome. 30,000. It's a quite small genome. For an example, if we compare this with our genome, human genome, it is 100,000 times than this. So in comparison to human genome, the COVID-19 viral genome is a simple, very small genome. Now we are going to look into this genome in further details. Basically, it contains 4 main parts. If we think this is the genome, COVID-19 genome, it contains 4 main parts. 1, 2, 3, 4. Out of these 4 main parts, the first part is dealing with the RNA polymerase, RNA dependent RNA polymerase, and this part acts upon the proliferation of the nucleic material inside the host cell. And the other part this is responsible for the protease production inside the host cell, and which divides the polyproteins into organelle of the virus. So this part of the genome is responsible for this job, and this part of the genome is responsible for RNA polymerase action. So what is the third part? This is the part which is more important for our discussion today. This part, the third part, is the part which is responsible for the spike protein production. And the last part is responsible for the nucleocapsid protein and the envelope protein production. Envelope protein production. Just to remind you, this is the genome which contains that 30,000 pattern of bases, 30,000. Out of that, the first part is this, second part is this. This is the third part which we are going to discuss about, and this is the part which is responsible for the spike protein mutation. Now, we are going to analyze this spike protein gene of the COVID-19 genome in view of its mutations, and discuss about the prominent variants all over the globe. Okay, this is the spike protein gene which I took out of the COVID-19 single-stranded genome. So in this spike protein gene, there were few mutations which are related to the new variants that we found all over the globe. Out of them, the first one was N501Y mutation. This is the one which was found in the UK variant, N501Y mutation. This was found in the UK variant B117, and this mutation causes a change in the receptor binding domain of the spike protein, by which it more facilitates the entry into the cell, host cell, by touching the ACE2 receptors. So this spike protein with this mutation become more attachable to the ACE2 receptor, by which the entry into the host cell is stronger than the wild variant. In this way, it became a very prominent variant, and the infectivity went up, and the viral load and the replication and viral production, by which the pathogenesis increased and it became a more difficult variant all over the globe, which was found in the UK for the first time, and it is known as N501Y mutation, which is in B117, the UK variant, and it is found in the South African variant B1351, and it was found in the Brazilian variant, which was called P1. So this mutation was found all of these variants: B117 UK variant, B1351 South African variant, P1 Brazilian variant. We have another two variants, the Californian variant and New York variant, in which this mutation was not found. The next mutation that I'm going to discuss now is the HV6970 deletion. This is a deletion of a particular pattern of these base molecules, and by which the shape of the S protein is altered, or the morphology of the shape S protein is altered into a certain extent. This was found in the UK variant HV69 and 70 deletion. This is a deletion of a amino acid sequence, and by which the morphology of the spike protein is altered. You know, the antibodies by the vaccine and the previous infection of COVID-19, the antibodies were produced against the spike protein. Now, this deleted, altered area of the spike protein is not attached to the antibodies. So, it is difficult, it is a bit difficult to destroy by the immune system. And still, there are other places where the antibodies can get attached and the complete spike protein is not changed, but there is a change and it affects the immune system. It affects the function of the immune antibodies in the process of controlling this COVID-19. So this deletion is also very important, change in the genome and the spike protein gene. The next mutation is E484K mutation. E484K mutation is also an alteration of the spike protein and by which it makes more attachable and more infective to the host cell. But this mutation is not in the UK variant, and it was found in the South African variant and the Brazilian variant. P618H mutation. P618H mutation. This is found in the UK variant, not in these two variants. And what it does is, when the spike protein gets attached to the ACE2 receptor, there is a protein in the membrane which facilitates the endocytosis of this spike protein and the virus into the host cell, and this P618H mutation, with the help of this membrane protein, it facilitates the engulf or the endocytosis process of this virus into the host cell, and thereby it causes more infectivity. So, as a result of these mutations, the infectivity of this COVID-19 is getting more and more, and by which the control of the COVID-19 infection is becoming a problem. On top of that, when they alter the morphology of these spike proteins, the immunity or the immunoglobulin that we made against the spike protein are not going to be much effective. For an example, the immunity, the immunoglobulin, the antibodies that we need to control this wild variant D614G original SARS-CoV-19 is in 6 times that we need to control this South African variant. For an example, if we want 1 antibody, now we need 6 antibodies to control the South African variant. 6 times we need the immunity against controlling South African variant when we compare the wild variant of COVID-19. That is why it has become a problem with the vaccination and passive immunization to control COVID-19. So I tried my best to explain the basis of these mutations and how the mutations happen and what are the effects of these mutations, and this is a very simple way of explanation, and this subject is very deep subject. I hope that you understood the basis of these mutations and the prevalent most prevalent mutations all over the globe now. I hope you gathered an understanding about the COVID-19 variants and their consequences and how they affect all over the globe. So if you have not subscribed to my Coffee Break channel yet, please subscribe and be vigilant about the next video, and I'm going to prepare another video for you in fortnight time, and you will be updated with another subject. Thank you so much for listening.